Thank you for inviting me today to speak to you about the experimental models of liver cancer that we use. And this really is a kind of joint session between myself and Chiara Braconi, who many of you will know from Glasgow. We are interested in really trying to expand the repertoire of models available for cholangiocarcinoma researchers, both kind of in vivo and in vitro. And really, we want this session to be very interactive. So I'm going to give you a short overview of the models that are available, and then we will have a long period after that in order to kind of discuss and kind of really kind of have a conversation about what the best models are out there. So what should an ideal model look like? Well, really, we think it should be patient-led, so it needs to reflect the pathology of disease, both at the histopathological level, but probably also the genetic level. These models should also kind of be able to be used to provide novel insights for either mechanisms, so how a tumor grows and forms, or to develop and prioritize new medications, and so for therapy. And really in this talk, I want to address the perhaps 2 or 3, I suppose, most kind of current advances in modeling, and really try and figure out whether these are reasonable tools to look at therapeutic testing. And there are a range of models that are available. So many of you will be familiar with these. The best model, I suppose, is the patient. And so understanding the cancer in the context of underlying disease, understanding the genetics of those cancers and the heterogeneity therein are important aspects of modeling. And because of things like high throughput sequencing, and also kind of new technologies becoming available in imaging, we can start to utilize the patient and patient data, particularly in understanding how to develop therapies for cholangiocarcinoma. Probably the most utilized model is 2D cell culture. So this is where cancer cells are grown in a dish or in a flask, and those have some obvious advantages. They're very cheap, but they're also very artificial. More recently, perhaps, we've kind of seen an increasing complexity in the in vitro tools we have available to investigate cholangiocarcinoma, and that has really come about from things like tumoroids and spheroids, which we'll discuss a bit more in the next few slides. More recently, again, precision-cut tissue slicers, where we can take patient samples and slice them and maintain them in culture for a while. And then those kind of in vitro tools move into the more basic in vivo models. So things like xenografts, where tumor cells are put under the skin of a mouse or into kind of the liver directly, and they can grow. There are obviously limitations there with the availability of an immune repertoire and whether those tumor transplantation assays actually reflect patient disease. And then there are the more complex genetically engineered mouse and fish models and rat models, which have been enormously useful both in cholangiocarcinoma research, but in other cancers. However, there are, of course, limitations. These are not patients, these are mice, and also they are astronomically expensive. So the limitation there is kind of whether they are available to everyone. So I will start in the next few slides just talking about these tumoroid cultures that have really kind of taken off over the last few years. And really, you can imagine organoids as tumors in a dish. So cells are isolated from the primary tumor mass. They're dissociated into individual pieces, and then they're seeded into a matrix. So this is normally a scaffold, which contains a number of growth factors, extracellular matrix, so these large kind of proteins that you find in the microenvironment of the tumor. And what we ultimately derive are clones of tumor cells that reflect the original kind of starting material, so in this case cholangiocarcinoma. And these have been enormously useful in research across the board. So from a number of different cancers, there are now substantial organoid libraries from varying patients with varying genetic backgrounds, various etiologies. So I think actually for the future, having these large panels of organoids is going to be very, very useful for discovery science. And really, my colleague in this session, Chiara Braconi, led a lot of the development of these organoids and actually demonstrated that they firstly recapitulate the primary tumor, but also they can be used to identify new biology and new therapeutics. And this really was kind of at the same time when Mary Hutch was also identifying similar organoids from both hepatocellular and cholangiocellular carcinomas. And really these two papers that I've kind of presented here were big steps forward in translating organoids into a kind of cholangiocarcinoma context. So what are the benefits, I guess? Well, the organoids that you kind of identify and grow and select for are maintained in a reasonably homogeneous culture media. And these organoids are, because of that, very, very consistent. So you get very, very, very reproducible data from culturing organoids, be it cholangiocarcinoma organoids or organoids from other cancers. They are also very reflective of the sample that you take. So a number of studies now have demonstrated that organoids kept in culture maintain their genetic identity. They don't become more mutant over time. So they really do reflect the early genetics of the cancer rather than becoming their own self-selecting population of cells. And you can see here there are different types of, I suppose, organoid or spheres that you can grow. So in the top, you can see a 2D primary cell line. So this is just cancer cells grown in a dish. When you aggregate those cells into a matrix, they form these spheroids, which are, they have 3 dimensions and they have some of the properties of a cancer, but they don't actually recapitulate very well the underlying tumor that they were derived from. Whereas 3D organoids absolutely look like the epithelium that you have isolated in order to establish these lines. And this is a lovely example here. So in the top panels from this Lampis et al. Paper, you can see the biopsy that was used to define these organoids or generate these organoids. And in the lower panels from that same paper, you can see what the epithelium of the organoid looks like. So actually, these things do recapitulate very nicely the kind of cell of origin, I suppose. And the same was kind of shown in the Braconi paper from Mary Hutch's lab, where the pathophysiology of disease is reflected in the organoid. And actually, these become very kind of useful predictors of patient response. So drugs that might work in these organoids to kill them might actually work in the patients to treat them. So these are really, really sensitive, kind of refined ways of doing drug discovery. So what are the benefits and pitfalls here? Well, so 2D cell lines are easy to grow, pretty cheap, with very high throughput. So you can really screen novel compounds in these, but actually they're not very robust. Actually, they can be batch to batch variable, translatability between a 2D cell culture system and the patient is incredibly poor. For patient-derived organoids and patient-derived xenografts, which is where these organoids are kind of taken and put into a mouse model, can be more time costly, but actually it's not that bad. It's weeks and months rather than years. They can be expensive, particularly in the case where these are going in vivo. Throughput is variable, but organoids, obviously, if you're growing them in a dish, is higher. And in both contexts, actually, patient-derived xenografts and patient-derived organoids have been shown to really recapitulate tumor sensitivity to compounds, so that actually their robustness is very good. The one limitation currently, I think, and I think it is a substantial limitation, is that organoids are removed from their microenvironment, and so they reflect the epithelium that the patient has in their tumor. They also only reflect really the epithelium that's contained within the biopsy. So understanding either tumor heterogeneity with organoids, or indeed kind of what the interaction is between the microenvironment and the cancer cell is quite limited if you're only using organoids. There are new techniques becoming available where cells can be added back to the organoids, so stromal cells and immune cells, but those are very much in their infancy and haven't really been shown to completely recapitulate the microenvironment in cholangiocarcinoma. So this is where I think things like genetically engineered mouse models and carcinogen models really come into their own, because what we're looking at there is the cancerous epithelium, but in the context of an immune system and a microenvironment. So the administration of thiacetamide to rats really has been around for a long time, and this is a very kind of, I suppose, large marker in the modeling field for cholangiocarcinoma. And this was published many years ago, 60, 70 years ago, where long-term administration of thiacetamide to rats gave them cholangiocarcinoma. And actually, strangely, thiacetamide was used to preserve oranges in kind of antiquity. So weirdly, when we've used this model before, we have to dilute the thiacetamide in orange juice because the rats don't like the taste of it. So, you know, we've gone from preserving our own oranges to kind of giving thiacetamide and orange juice to rats. But I think the use of a thiacetamide-based or compound-based model has had its day in some ways. It's not very refined. It's quite protracted. It's incredibly expensive. So now many other labs are using making use, I guess, of these models called GEM models, these genetically engineered mouse models. And most of them are based on a technology called CRELOX-P. So CRELOX-P allows you to delete specific genes in specific cell lineages. So using a CRE, essentially as a molecular pair of scissors, you can chop out genes that are flanked by these sites called LOX-P. And this allows you to delete tumor suppressors such as P53 and P10, but also allows you, if you have the right mouse, to overexpress certain genes. So GFP, for example, so you can lineage trace and tag your cancers, but also allows you to overexpress candidate oncogenes. So one of the more popular ones, of course, is the overexpression of KRAS mutants in a lineage-specific way. And these have been an incredibly useful in order to try and understand what is going on in tumor initiation, but also in kind of tumor propagation and the interaction between the cancer cells and the microenvironment in that situation. The limitation, of course, here is that actually you are selecting for a very restricted number of genes to look at. So you are basing your model around a particular genotype of the cancer. And that actually might hinder your therapeutic discovery. And really kind of like lastly, I guess, the adaptation of in vivo models has occurred over the last few years, and that is this hydrodynamic model. And I don't think this is separate to the GEM models at all. I think GEM models are excellent, but also this hydrodynamic model is being utilized more and more. And the idea here is that you can deliver naked DNA intravenously under high pressure, so you inject through the tail vein, and the DNA cargo is essentially transposed into the liver. So you use a genetic trick where you express a transposase, flank your genetic cargo in a transposon, and once that's in a cell, it essentially gets shunted into the genome. So this doesn't rely on classical animal crossing that you require for GEMs, so it's actually very efficient. You can do it in wild type mice. These systems are incredibly modular, so you can express certain oncogenes, remove oncogenes, put new oncogenes in, so they're very flexible, and actually they're relatively cheap to generate. And with the addition of CRISPR technology, you can actually use this method not only to address what happens in one gene, but also what happens in a panel of genes. And I thought I would just put this video up because it's very interesting. When you inject a mouse with, this is contrast, very slowly, you can see that the contrast just kind of goes through the mouse's vasculature. Whereas when you inject hydrodynamically, so this is under high hydrodynamic pressure, the cardiac capacity is overwhelmed and you get backflow of the contrast into the liver. And this is the trick that is used in this system in order to deliver these DNA vectors into the mouse and ultimately those initiate tumors. And as I mentioned before, you can use this to generate all types of cancer. And in this paper here, just as an example from Holger Willenbring's lab, they've utilized this to generate cholangiocarcinomas. And you can see over time that these tumors initiate and they grow and they grow and they grow, ultimately until they overtake the tissue. Of course, one of the questions here is cell of origin and whether actually these tumors recapitulate the cell of origin in patients. And I think that's something that perhaps we can discuss in the session afterwards. And as I've previously mentioned, using CRISPR in this system is incredibly powerful because you can not only delete or affect one gene, but you can actually delete and affect multiple genes. The thing that has come out of human genome and human exome studies in cholangiocarcinoma is that these are very, very heterogeneous cancers genetically. So understanding how that variation in the genome and in the mutational profiles affects tumor progression, I think is going to be very, very important for developing new therapies and trying to ascertain whether certain treatments will work in certain patients. And we now have the tools, I think, in order to address that. So what are our kind of big conclusions, I guess? Well, understanding the genetic complexity of disease is challenging, but using tissue from patients to build models is important because then they reflect pathology. You can use those to ask fundamental questions, you can use those to use translational questions, and organoids really kind of help with that. But if you need to add complexity to a system, then you still need to go in vivo. And you can use these animal models to generate tumors. And I think actually that improved animal modeling that more closely reflects the patient will be important to test the causative events in cancer, so the genetics of cancer, but also understand new therapeutics. So with that, thank you very much. And I will open the floor, I guess, to discussion.
Welcome for a final time. Following our last presentation of the day, we're now going directly into a live discussion and Q&A with Dr. Luke Boulter and Dr. Chiara Braconi. They're now here to answer your questions, as well as facilitate a discussion between themselves on Luke's recent presentation. So to start off with this session, I'd like to hand over to Chiara, who will start us off.
Thank you very much, Gary, and thank you, Luke. It was really a very great presentation and a nice overview of the topic on experimental models where we currently are. Where do you think, as a first question, before we wait for the audience to kick in questions, where do you think we need to go in terms of experimental modeling? We have seen very nice data about intrahepatic cholangiocarcinoma and in vivo modeling. Do you think we need to extend a little bit more on other subtypes of cholangiocarcinoma, and which are the challenges of developing animal models for the other subtypes?
That's a good question. And I think what we've seen over the last maybe decade or so is that we have had an expansion in models, and certainly with organoids and the GEMS and the hydrodynamic injection models, we are expanding our range of tools. You're right, though, there is a very large bias towards intrahepatics, and there are very few models to look at extrahepatic cancer. So I think there's definitely a requirement for the development of some really useful and kind of tractable extrahepatic cancer models. The way I see perhaps the slightly more developed field of intrahepatic modeling going is that we need to reflect probably the genetic diversity of these tumors a bit more. We still rely very much on the deletion of P53 and then thiacetamide or injury. And we probably also need to understand whether our cancer models actually really do reflect pathology. I'm not always convinced that they do. And I know many of us on this kind of, in this area have had a discussion about whether and how we kind of correlate our animal models with patient data. Like I said in my talk, though, the one thing I do think that we really get out of that is we can look at these interactions between the kind of genetics and also the microenvironment or the kind of inflammatory processes that happen there.
On that note, actually, there is a question from the audience. So Ed is asking, congratulating on a talk, and then he's asking if it would be clearer, a clearer stratification of immune subtypes in patients help the development of more targeted in vivo model. And what would genetic modeling of different immune contexts be as a future possibility?
I think that's certainly a future possibility. And I reckon that something like the hydrodynamic model really lends itself to that, where you can put in very stereotyped mutations into the epithelium and look at how that influences the immune microenvironment. But I think, kind of like the work you've been doing, Chiara, with Jesper, is really kind of like leading into this idea of immune stratification and understanding these subtypes. I mean, I'd be interested to hear what you thought on whether you see that as a real kind of step forward in how we treat patients.
Well, definitely organoid might be one of the way. I think we are still at a very preliminary stage, at least in cholangiocarcinoma. I think that it's a good idea, and that would be, a promising avenue that we can derive mini tumors from the patients and then bring them in the lab and test them on different drugs and potentially give the patients the correct drug. I think before we move there are still some questions that we need to answer, especially in the biliary cancer field. As I said before, first of all, I don't think we still have a clear idea of which is the efficiency rate of organoid development in all different three subtypes of cholangiocarcinoma, especially if we move away from the resected specimen, if we go into biopsy and collection of fresh tissue there. I think it's also important to consider that compared to other forms of cancers, for example, colorectal, where clinical trials are a little bit more advanced and they are now incorporating organoids into, individualization of treatment, we need to acknowledge that biliary cancer is still a rare cancer. So most of the time we need to bring together a lot of different centers to really have high numbers to make an impact in changing clinical practice. And with that in mind, I think that we need to overcome some logistical issues of deriving, organoid and establishing organoid from the fresh tissue. As as a clinician, I also appreciated the difficulty of doing a biopsy and then shipping straight away to another laboratory. So I think that we needed to overcome logistical issues there. For example, deriving protocols to establish organoids from cryopreserved tissue that facilitated the situation. I think that on that background, I think that organoid is a promising tool, but it's not probably the only one. And organoid, as we said before, has only an epithelial component. So if we wanted to move ahead from testing chemotherapy drugs or anti-proliferative compounds, then we need really to move on to greater and more complicated platforms, like, for example, organoid on chips or precision cut liver slices, which are definitely another good model where you have the microenvironment there, and then you have the potential to test also immunotherapeutic drugs. With the flow on that side, that then you can't culture them for too long and for a long time.
So I guess there's a question that I will lead on to from Kevin Gaston, but I'm going to kind of preface it with another question. So, when we biopsy people and taking these very small cores to grow organoids from, obviously that only represents a single clone within that tumor. And we know that when we take kind of bulk tumor to sequence, that there's a very diverse genetic profile in that cancer. Do you have any concerns that by being so reductionist in our kind of sampling that that you're essentially going to kind of miss a lot of the biology that's in that tumor already?
Well, for definitely that is going to be a limitation on organoids, because we are culturing what we pick up, and definitely if there is an intratumor heterogeneity in the tumor, then we won't be able to reproduce that unless we do multiple biopsies. But I think that is something that brings us to another level of error that is embedded in our clinical practice anyway. Even when we do a biopsy for diagnostic purposes, we are just looking at what we're sampling.
Yep.
And for example, I'm thinking about this issue, mixed forms of HCC and cholangiocarcinoma. I think this is becoming more and more frequent. At least in my practice, I tend to see more and more this mixed form. And coming back to your expertise on cell of origin, I see that, on this background of cirrhotic liver, now these tumors arise altogether. How would you comment then from your biological point of view and your expertise there? Do you really think it's the same cell type that is going to move forward into two different patterns of tumors?
I mean, like, it's a question I struggle with, and I think it's an interesting one. That so we heard talks earlier today about the kind of effect of or the potential for kind of progenitor cells in the liver to be the precursors of cholangiocarcinoma and HCC. And I certainly think that's one option. I mean, I think increasingly, though, what we recognize, at least in mouse models, is that there's a huge level of plasticity within these two lineages. And even actually things like the hydrodynamic tail vein injection model relies on that lineage plasticity of the hepatocyte in order to generate these cholangiocarcinomas. So what the actual cell of origin is for a cholangiocarcinoma, I would still like to think it's a cholangiocyte. And I think in the majority of cases it is. But that's not to say that hepatocytes can't undergo this kind of transdifferentiation event, or that progenitor cells can't essentially become cancer stem cells and feed into both. I think the jury is very much out, though. It's a very interesting set of questions to ask.
Yeah.
So one of the things that that Kevin wanted to know was why do your organoids not continue to mutate in culture? Since within a person, you evolve this intratumoral heterogeneity. Why do you think that stops in vitro?
Well, in vitro, we don't see that the organoids change the mutational pattern that much. I mean, the driver mutations continue to be always the same. From a transcriptomic point of view, it is maintained, but logically it doesn't remain the 100% of what it was originally. I think we have to be careful when we say that the tumor changes so much, because I guess that there are some nice data from Johns Hopkins, which really identified how the genetic background of tumors do not change that much unless they are exposed to chemotherapy and treatment. And I think that is the other open question that we still have before we move organoids into clinical practice. So what happens after you treat them with chemotherapy? One of the ideas we had was to really expose them to chemotherapy drugs and see if we can make them resistant so that we can try to mimic what happens in the human patients. But definitely we will miss all the rest of the component of the tissue microenvironment, so that would be limited to specific class of compounds. Answering one of Anna Gavroska questions about if we have done this yet, not in cholangiocarcinoma organoids, we haven't really seen, we haven't managed to do a chemotherapy-resistant organoid line yet. And we haven't really gone in depth into the analysis of single cell analysis and what happens when you treat them with chemotherapy. This is something on the pipeline. We have done for other tumor types, but we haven't done for cholangiocarcinoma. We tend to see, for example, with cholangiocarcinoma organoids, and I'm very happy to hear the thoughts of other people that can feed in the chat and in the questions if they have experience. We have seen that these organoids have kind of phases of growth. They tend to grow very nicely, and then they have kind of a plateau, and then they re-pick up. And I don't know if anyone has noticed this, and if there is any interaction with extracellular matrix and all the support that provides as well. And I think that also it's important what comes from the rest of the organ as well. And this is why animal models maintain and they will continue to maintain their role. Try to answering Meg Illingworth's question, for example, about how, what are the models out there to try and reflect risk factors and PSC. Do you want to comment on that, Luke?
Yeah, sure. So actually, it's a complicated area. So the original models, kind of based on carcinogens like diacetamide, actually give you an inflammation and fibrosis that ultimately, over quite a long period of time, evolves, I guess, into a a tumor. So in that regard, you have this kind of pre-neoplastic state that transitions into a cancer. So it's quite a good model, but it's very long. And this is definitely a kind of area close to my heart. So we're trying to generate new models where we can accelerate that. Because I think, you know, the question about how pathology and pre-malignant disease interfaces with tumor formation is really important. So do you require those pro-regenerative or pro-repair signals that you see in kind of pre-malignant disease in order to help initiate a cancer? And actually, we've got a kind of research program looking at that at the moment. But it's complex because you have all the underlying disease pathology as well. But hopefully watch this space and we might have some progress soon.
And actually, my thought on PSC is that probably the animal modeling will be better served by animal modeling rather than by organoids. As you know, genetically, we have seen from some published data that genetic landscape of PSC-related cholangiocarcinoma are very similar to extrahepatic cholangiocarcinoma. So I think it's the inflammatory background that needs to be mimicked there.
And I think that links nicely into one thing that Toby Fessy is asking about whether there are any kind of models that develop METs, because I think my feeling is that, you know, kind of when you can start to model these microenvironments, then you'll be able to look at both the kind of genetics of the primary tumor versus the metastasis, but also what are the changes that are necessary for those circulating tumor cells to disseminate from the primary cancer and kind of move off elsewhere. As far as I know, in animal modeling at least, a lot of those primary tumors are terminal before they metastasize. There are a couple that I think Diego Carvesi is testing out at the moment, which might actually kind of be quite promising in modeling metastasis. But generally, yeah, it's a primary tumor kind of phenotype we see at the moment.
And definitely from an organoid or non-in vivo modeling, this would be a very interesting question to answer. I think the problem is if we wanted to create patients-derived models, is that then there will be the flow of going and biopsying, different sites, metastatic sites. So that becomes much more harder for the patients themselves. So probably there might be a way to overcome these issues with autopsy programs, but it will be more challenging to reproduce that in vitro as well. And I think there was a question by Colm for you, Luke. That's a very challenging one. And what, you have talked about some animal models induced by exogenous insults, like for example, DEN. How do you think they are relevant in the clinical practice, as these are very typical of mice and usually we don't tend to see this signature in humans? So should we completely give up on those models, or what role they would still have?
It is, and I don't want to upset a large portion of the audience either. So I think the things like DEN, I think they have a place. I don't use them. We do use diacetamide, we don't use DEN. Because for exactly this, you don't really see the mutational profiles that you see in patients using those types of approaches. That said, as a mechanism by which you can get something that pathologically or histopathologically looks like a cancer, or a cholangiocarcinoma, they can be quite useful. And there's a bit of me that's kind of moving towards the idea more and more as cancer as a disease of signaling rather than as a disease of genetics. And so almost whether you have the right genetic profile may be inconsequential, as long as you've got the right transcription on the right proteomic profile for you to be a cholangiocarcinoma. Now that might be quite topical. So in that regard, I think if you're wanting to understand the cell biology and the kind of pathophysiology of a tumor, then they're acceptable tools. But that you have to accept the caveats. And I think wherever we are with cell lines or organoids or animal models or PDOs, all of these things are experimental tools. And I think if you rely on one too heavily, then you're essentially just testing the ability of that model to respond to your therapeutic or to kind of answer your biological question. And so I would encourage people to use a range of tools for which DEN may be appropriate.
Well, actually, I was thinking exactly the same, reading the question by Shireen, where she's asking which big data sets would help to convince the field that in silico models of patients may be the future. I think that probably we just need to think that is an integration of different models. We won't have one single model to give the complete answer. And there is another question from Toby, which is very interesting, about the current GEMS model that develop. Are you aware of any model that develop a metastasis from the primary? And does this require too many alleles to be practical, or do you think that we can get there? And with regards to that, I have another question following, because I think that one of the problems of GEMS is that, as Toby is pointing out, you can't add too many, hits. Would your method of hydrodynamic tail vein injection be a way to overcome this? How many genes can you modulate?
Yeah, so I mean, like in our library that Molly presented earlier, we targeted 91 different genes with multiple different guide RNAs. So you can certainly multiplex in quite a high level in that system. So practically, yes, you can do that. I think the thing will be to identify the mutations that are necessary for metastasis, and then you can add those into your kind of hydrodynamic method. We've got some evidence that mutations in certain genes like NCOR1 might be able to promote metastasis, and we've not really followed that up yet. But certainly, yeah, I think the hydrodynamic model would be a good opportunity for that, particularly because you say actually once you've got to kind of 4 floxed alleles in a gem, really your kind of the rate of production of useful genetically engineered mice is so low in breeding that it's almost impossible to add more.
So I'm sorry to have to interrupt you both there, Chiara and Luke. That conversation could clearly go on for many more hours. But I think it's a fascinating conversation to end on, and I think one that really demonstrates the importance of the conference to have this level of conversation at 5 o'clock on a Friday, with more questions to go. I think is a testament to the work that you're both doing, and everyone that's contributed towards the conference this year. So thank you, Chiara. Thank you, Luke, for participating in the conference.